Welcome back to Chapter 21, everyone. This is going to be Part 3. We are going to continue our discussion of the adaptive immune system. And what we're going to do is we're going to go ahead and take a look at our T cells in a little bit more detail. Take a look at the different versions of them, which one are effectors, and which ones will help us form our memory of the interaction with the foreign antigen. Now, as a quick reminder, your T cells are part of the cellular immune response for our specific response or our adaptive immune system, and that is because it's going to have an intracellular reaction, and the T cells are going to be able to directly kill the foreign antigen. T cells, because they're able to do this and directly kill the foreign antigen, will be a little bit more complex than our B cells. Quick reminder, our B cells are the ones that will form the plasma cells, which will secrete the antibodies that can tag and immobilize the foreign antigen, not destroy it. And our B cells are part of the humoral immune response. Now, within the T cells, what we see happening is that there are two main populations. And this is all based on the fact that they have differences in the receptors that are on the surface of these lymphocyte cells. And what we see happening is that you can either have what we call a CD4 cell, or on the next slide, we'll talk about a CD8 cell. The CD4 cells are naive versions of the T cells that will eventually become helper T cell. Helper T cells get their name from the fact that they will assist or help activate other B cells as well as T cells and other innate cells such as our macrophages. They are going to be almost like the assistant or the coordinator that will connect the innate and the specific immune system together due to the fact that they can help and they can recruit additional cells over to the site of injury. Now, in addition to the helper T cells, your CD4 cells can also go ahead and mature into what we like to call suppressor T cells. Some books like to call them regulatory T cells, as well as memory T cell. So a CD4 cell has the potential to become a helper T cell, a suppressor T cell, or a memory T cell. In addition to the CD4 cell, you also have a CD8 cell. Now remember, they're named for the fact that they have differences in their glycoprotein receptors on the cell surface. Your CD8 cells have the ability to mature and become your cytotoxic T cells, also known as your killer T cells. The killer T cells get their name because they are the ones that will actively destroy or kill the foreign antigen. Your CD8 cells also have the ability to become memory cell. Now, the memory cells you'll see in both CD4 and CD8, because it is an intricate part of the adaptive immune system, is the fact, obviously, to create a memory of the attack. Now, just so you're certain, just so that we're certain, I guess, that everybody understands, when you see the mention of CD4 and CD8, we are still looking at what we call naive T cells, meaning they're not fully mature yet. The CD4 cell will become the helper, the suppressor, and the memory T cell, where the CD8 cell will become the cytotoxic or killer T cell, as well as a memory cell. As we continue our lecture on part 3, we'll come across a lot more differences between these two. So for now, it's very important to know the difference between the naive CD4 and the naive CD8 T cell. Here is a quick illustration that will highlight the discrepancy between them. So as you can see, we are looking at our immature lymphocyte. It will travel over to the thymus because the thymus is where we're going to have our positive and our negative selection so that we can make our T cells immunocompetent. And that is then also where our immunocompetent T cells will be divided into CD4 and CD8 cells. And the CD4 cells will be the ones that will eventually give rise to our helper T cell, our regulator or suppressor T cell, as well as obviously our memory cells. And another difference that we see here is what is actually going to go ahead and activate this immunocompetent cell? What is going to allow it to mature? So what's involved in its activation? And for the CD4 cell, it's going to mean that it's going to have to interact with an antigen-presenting cell, like our little dendritic cell right here. It's going to have to interact with the class 2 MHC protein. MHC was briefly mentioned in our previous part. It stands for Major Histocompatibility Complex. It is a protein that can house an antigen. And in a little bit, we're going to talk about how you have a class 2 and you have a class 1 MHC. The class 2 MHC will be the one that interacts with the CD4 cell to allow it to become activated and eventually either become a helper T cell, a suppressor or regulatory T cell, or a memory T cell. If you look over to your right-hand side of the illustration, you're going to notice that your CD8 cells will also interact with the antigen-presenting cell, but they will interact with the class 1 MHC protein, and its interaction will allow the CD8 cells to become activated and allow it to ultimately become the cytotoxic or killer T cell, as well as, of course, the memory cell. Okay, so CD4 interacts with class 2 MHC and becomes a helper, a regulatory, or a memory T cell. CD8 interacts with class 1 MHC and will become your cytotoxic or your memory T cell. Now, what exactly is this MHC? It stands for our Major Histocompatibility Complex. It is a group of proteins that will be able to display the little fragments of the antigens. Now, these antigens obviously are going to be foreign antigens because it's what's going to be activating our immune system. We need to have activation or exposure to these foreign antigens before the T cells can go from naive, meaning triggered as labeled as CD4 and CD8, to being fully activated. And that's then the point where you will go ahead and have the effector cells, the killer T cell, the suppressor T cell, the helper T cell, and of course also the memory T cell, which is so important for maintaining our immunity and being part of our adaptive immune system. Here is a little bit of a closer look at the differences between class 1 and class 2 MHCs. Class 1 MHC proteins will be displayed on every single one of our cells, with the exception of the red blood cells. Class 2 MHCs will only be displayed on antigen-presenting cells. So those are going to be your dendritic cells, your macrophages, as well as your B cells. Now go ahead and take a look at the illustration and it will show you the importance of your MHC protein. Over here on the illustration, you have a phagocytotic cell. As you can see, it's able to do endocytosis on a foreign antigen. And you've seen a similar picture like this before in part 1, where the foreign entity is able to be engulfed inside the cell. We form the little vesicle, and then we expose it to different lysogenic enzymes. And in this case, let's assume this is a bacteria. As the bacteria is degraded and destroyed, we can get rid of some of the fragments by kicking it out. But also look what happens here in step number 6. We take a little piece of the foreign antigen, and it's then displayed over here in this little blue structure. This little blue structure, this little housing mechanism, this is your MHC protein. And the foreign antigen will sit in here on display. It's labeled as the epitope because that's going to be the active part of the antigen. And then what we see happening is if the epitope or if the antigen is on a class 1 MHC, it will be able to interact and be recognized by your CD8 cells. If it's on a class 2 MHC, it's going to be able to be recognized on a antigen presenting cells. Okay, so all of your cells will have class 1, and then in addition to that, your antigen presenting cells will also have class 2 MHC proteins. So when you look at your dendritic cell, when you look at your macrophages, when you look at your B cells, they will both have class 1 as well as class 2 MHCs. Here is a look at the fact that your antigen-presenting cell, like your dendritic cell, has both classes of major histocompatibility complexes, as I was mentioning to you on your previous slide. So here's my little dendritic cell, and notice that they went ahead and they drew for you the class 1. That's the one that all cells have except for our red blood cells. And notice how the class 1 is the one that can interact with the naive CD8 cells, which will eventually mature into the cytotoxic or killer T cells, as well as memory cells. Their other MHC, class 2, here on the left-hand side, is the one that will cause interaction between the CD4 cells, and the CD4 naive cell can then go ahead and develop into the helper T cell, but also the suppressor T cell, also called the regulatory T cell, as well as a memory T cell. Here is a nice look at the comparison between class 1 and class 2. So as you can see, for class 1, it says that it's displayed on all nucleated cells within our body. That's why it excludes the red blood cells, because mature red blood cells lose their nucleus, as you might know. And they're going to be able to be recognized by our naive CD8 cells, as well as their mature version, which is going to be the cytotoxic T cell. The class 2 MHC proteins, those you're going to find only on your antigen presenting cells, so your dendritic cells, your macrophages, and your B cells, and they will allow interaction with the naive CD4 cell as well as the helper T cell. All right? So anytime you see class 1, I want you to think CD8, which eventually is going to lead you to the cytotoxic or killer T cell. Class 2 is going to lead you to the CD4, which means it's going to eventually develop into a helper T cell. And once again, MHC, major histocompatibility complex, it is a protein complex that allows you to display antigens. Here is one more picture. I just want to make sure that I keep highlighting the fact that in order for us to get to our effector cells of our cytotoxic killer T cell and our helper T cell, these cells have to have an interaction with the MHC proteins. So if it's class 1, that one will be recognized by your CD8 cells, which will become your cytotoxic killer T cell. And if it's class 2, we will only find those on antigen-presenting cells, and that will cause activation of your CD4 cells to go ahead and develop into helper T cells. Alrighty, now let's take a little bit of time and talk about the actual interaction that's occurring between the MHCs and your naive cells. In order for you to fully activate your T cells, and in essence what you're doing is you're priming them, so you're showcasing this particular antigen because you're looking for a very specific response. In order to activate the T cells, we see that there is a two-step process that needs to be done. We need to get antigen binding completed, which is going to be the physical interaction between the antigen and the naive T cell. And then we're going to need to have co-stimulation. And that's almost like a double check. We're going to have to make sure that we are indeed recognizing a foreign entity. Both of these will occur on the same antigen presenting cell, and both of them are required for clonal selection of the T cell. And what that means is basically once the cell has recognized the foreign entity, that T cell that becomes primed, that becomes activated, will go ahead and make lots of clonal copies of itself, so it can increase its numbers and thereby have a more amplified response as it goes off to fight the foreign entity that was presented through the MHCs. So what exactly is involved with our first step of activation, which is the antigen binding? Well, let's go ahead and see what the PowerPoint tells us. So it says right here, it says you have these T cell antigen receptors which will have to bind to the MHC complex on your antigen presenting cells. And when they interact, we're looking for a double recognition. And the double recognition basically means that your naive T cell has to both recognize the MHC complex, so it has to recognize the protein, and it also has to interact with the foreign antigen. All right, so it's going to go ahead and bind in a way that it's going to allow for interaction between the MHC as well as for the foreign antigen. Now, obviously, along the way, there will be some other pathways that are activated, some other proteins that might be needed. But at the core, the antigen binding literally just means that we're having recognition and physical contact between the MHC, the foreign antigen, and your naive T cell. In order to make sure that you require a T cell so that the antigen is foreign, in addition to the antigen binding step, we're also going to do co-stimulation. And co-stimulation is going to rely on co-stimulatory signals to basically confirm the recognition of the foreign antigen. This is extremely important because it turns out that if we don't have co-stimulation, you can have allergy that can occur, which basically means that you have your immune cells that can become tolerant to an antigen, that they're not able to divide, and that they're not able to secrete cytokines. Cytokines are chemical messengers that can be used to amplify the signal as well as recruit additional immune cells over to the site of injury. So you need this two-step process of antigen detection and co-stimulation to make sure that you are properly activating your T cells, and that of course will limit our chances of developing a cross-reaction or like an autoimmune disease. Once we've completed the two-step process that's required for our T cells activation, we will see that our CD4 and our CD8 cells will go ahead and start to mature, and as they become primed, we'll see that the T cells will undergo proliferation as well as differentiation. Now, along the way, we see that the proliferation will mean that it will increase the number of T cells that will be able to respond to the foreign entity. Usually it takes about 7 days for the T cell response to peak to its highest, meaning it's actively fighting off the foreign antigen. And after that, we will need the numbers to start decreasing because as the antigen starts to disappear, we also want to get rid of our activated T cells that we no longer need because our immune system has done the job that it's set out to do. Now, part of the reason that we want to go ahead and taper down our T cell response is because as the T cell is inducing apoptosis and killing off our foreign antigen, it also sets off another type of response, which means that it's going to cause inflammation to occur. So it's almost like there's a site of injury that's occurring. And if that site of injury isn't tapered down and cleaned up quickly, it can actually increase our chances of having hyperplasia, which is that our cells become enlarged and stop functioning normally, or we can have, even worse, we can have cancer cells that start to make an appearance. This, of course, can put the body at a disadvantage. So what we see happening is that we want to make sure that the killer T cell solely focus on the foreign antigen, and once those are done, we want to go ahead and get rid of them if we no longer need them, because they could be hazardous and cause a secondary set of symptoms to appear. Now keep in mind it's okay to get rid of your killer or cytotoxic T cells because you have your memory T cells that will keep track of the infection and will be able to respond in case there is a secondary or sequential exposure to the same antigen. Here is a nice little look at our activation of our adaptive defense system. So as you can see right here, we have our antigen presenting cell. Here's my little dendritic cell. I have a bacterial antigen that is going to be interacting with, and as it's doing its phagocytotic mechanism, it will also take a little piece of the bacterial antigen, and right over here you can see it's going to display it in its class 2 MHC complex. So once again, the MHC complex is the purple structure, and then the little red staff right here, that is your foreign antigen, your little bacterial antigen. So on my chart it says we have antigen presentation. The dendritic cell has engulfed an exsanguinous antigen, meaning something that doesn't belong, and it will display its fragments on a class 2 MHC protein. Now remind yourself that class 2 is going to be specific for the activation of our CD4 naive cell. So what we see happening is that the activation is going to involve a double recognition because our CD4 cell, right over here, needs to interact with both the MHC here in purple as well as with the foreign antigen. And in addition to that, once we have our double recognition, we also need to have a co-stimulatory molecule. So you can see the little receptor interacting right here, almost forming like a little bridge format. That's our co-stimulatory molecule receptor. And that in essence is double checking our initial interaction. Once these two have occurred, so we have our double recognition and we have our co-stimulatory response, we then see that our CD4 cells will go ahead and transition into a helper T cell, and when it does so, it will proliferate. So notice how you went from just one cell to 4 cells. And this is an example of clonal formation because you're literally making copies of the same cell. And notice how the majority of them will become helper T cells. And then you will also have some memory T cells that will stay intact for the next time you become exposed to the same antigen. Your cytotoxic T cell will do the same exact thing, but obviously with the exception that it will interact with the class 1 MHC. It will have the same exact reaction that it needs to do with the double recognition, and it will go ahead and also do the clonal formation. In fact, in a little bit, we're going to talk about the fact that the co-stimulatory molecule for your CD8 cell will often come from the CD4 cell because your helper T cell is an integral, very important part of our immune system. Now, the term cytokines, you're going to see that quite a bit as well. These are chemical messengers that your different immune cells can secrete and use as a mechanism to communicate between each other. Within cytokines, you will find your interferons, which were the messengers, the protein messengers we discussed that virus-infected cells will secrete. But you will also find your interleukins. Interleukins are released by your macrophages, and they are an excellent way to kind of alert other white blood cells and recruit them over, including our B cells and our T cells. So these are your chemical messengers. All right, so let's go ahead and take a look at just a little bit more detail about what each of these specific T cells brings to our cellular immune response. So we've discussed the helper T cell, and I mentioned before that it's all in the name, that it will help recruit additional B cells. It will be able to communicate with the killer T cell. It will even able to communicate with macrophages, other antigen presenting cells, as well as some of your innate cells. Because it's able to interact with both the humoral, cellular, and innate response, your helper T cell is probably the most important immune cell that you have in your body. And that is because it's able to coordinate a reaction between all the different cell types and immune responses. So literally, without the helper T cell, there would be no immune response. So if you ever come across a question that'll ask you, what's the most important cell that we have in our immune system? Please make sure to look for an answer that's going to give credit to the helper T cell. Because as you can see right here, it can activate both B cells and other T cells. It can induce those cells to proliferate, so increase their numbers. And then it can also release those chemical messengers, those cytokines, to recruit over other immune cells, including some of your innate cells. So it's an excellent way of connecting all the different aspects of our immune system. So make sure to thank your helper T cells today. I also want to take a moment to talk about exactly how the helper T cell can activate the B cell. I know that we've discussed our B cells already in part 2. We talked about the fact how in order for them to be primed, they need to be exposed to a foreign antigen so that they can go ahead and differentiate into their plasma cells, which will be the antibodies, as well as the memory B cells. Well, what we see happening is that in order to fully maximize their activation, it turns out that the helper T cell will need to become involved because it will be able to communicate directly with the B cells through their MHC class 2 receptors. Because remember, class 2 is only for antigen presenting cells, which a B cell is as well. Once you have that interaction between the helper T cell and the B cell, we see that the B cell will divide at a much more quicker rate and antibodies will be formed in an accelerated format. Now, is it possible to activate your B cells without your helper T cell? Yes, and that can be done through binding of the T cell independent antigen, but it turns out that if you remove the helper T cell from the B cell activation path, the response will be very weak and it will be relatively short-lived. So the majority of the time you will have co-stimulation, meaning that there will be an interaction between the helper T cell and your B cell to maximize your antibody production. So here is a little illustration of that. Go ahead and take a look at the left-hand side, and it shows you how your helper T cell will help in the humoral immunity system. So that's the production of the antibodies. And in my illustration, I see my little B cell right here. And as you can see, it's class 2 MHC. It's displaying an antigen, which is interacting with my naive CD4 cell. And my helper T cell, which will eventually become, we see that it can bind to the MHC and the foreign antigen. And look what's going to happen. Your helper T cell, as it interacts with the MHC complex, is going to start releasing interleukins. These are these little yellow dots right here. And these interleukins are going to have a very strong co-stimulatory effect on the B cells because it's going to allow the B cells to amplify or speed up the production of their antibodies through their plasma cells. So your helper T cells can help the humoral immune system because it allows you to interact with the B cells, thereby speeding up their activation and their production of their antibodies. Your helper T cells also play a role in the activation of your CD8 cells. So they will also have the ability to recruit over and activate your CD8 cells, which obviously become your killer or your cytotoxic T cells. So in this case, when you have an interaction between your helper T cells and the CD8 cells, that interaction is going to be through the expression of co-stimulatory molecules. So this is our secondary checkpoint to make sure that our CD8 will truly need to be activated and the production of the cytotoxic killer T cells is essential for your immune system. So let's take a look at that illustration. So here's the same figure we saw previously, but now we're looking on the right-hand side, and we're taking a look at how the helper T cell is interacting with the cellular immune system. So once again, we have our little helper T cell. And then notice here how the helper T cell is making its recognition and its antigen binding with its class 2 MHC, which we expect. And we have our little antigen presenting cells right over here. Here's my little dendritic cell. So we expect it to display its class 2 for the helper T cell. And then below we see that it has its class 1 that's fully interacting with our CD8 cell. Right? So that's the one that's going to become our cytotoxic killer T cell. And remember, we talked about the fact that it's going to need to have a co-stimulatory molecule. And what it says right here, it says your helper T cell will bind to your dendritic cell. That's the antigen presenting cell. And then it says the helper T cell will stimulate the dendritic cell to express the co-stimulatory molecule. So here we go. That's the double check to make sure that everything's okay. We need that costimulatory molecule. And then the dendritic cell can now activate the CD8 cell with the help of the interleukins, here in yellow, that are being secreted by your helper T cell. And the addition of the interleukin is going to speed up the process of the naive CD8 cell converting and proliferating into a cytotoxic or killer T cell. So this picture is extremely important because it highlights the connection between the humoral and the cellular immune system through the assistance of your helper T cells. Now, your cytotoxic / killer T cells, these are the ones that can directly attack or kill the cells. They are, once activated, will circulate through the blood and the lymph. No, it's not a surprise to anybody. And their targets can be a wide range of things. We can have virus-infected cells, cells that have intracellular bacteria or parasites, cancer cells, as well as really any type of foreign cell that might be introduced to the body. Your cytotoxic T cell will deliver its lethal hit very similar to a natural killer cell. It is going to use perforins and granzymes to literally poke a hole in the cell membrane of the foreign cell and induce apoptosis in this foreign entity and get rid of it that way. So on your slide, I was sure to mention that it has the same mechanism as the natural killer cell, through the use of perforins and granzymes, and the whole goal is to induce apoptosis in the foreign entity. So let's go ahead and let's take a look at a picture of that, or an illustration, I should say. Here you go. So here's my cytotoxic T cell. It will go ahead and interact with the target cell. Now remember, your cytotoxic T cell checks MHC class 1 proteins. As it scans it, it's able to see if it's foreign or not. If it's foreign, it will go ahead and induce the release of perforins. Perforins will make perforations, little holes, inside of the cytoplasm of the foreign cell. And once the perforin pores have been formed, it's then going to release granzymes, and granzymes will be able to enter into the target cells, and they will cause the target cell to induce apoptosis, meaning that it will start fragmenting from the inside. It will start imploding, and that target cell will soon be dead. We call it cell suicide. And then like a macrophage will come by and engulf it, and off it goes back to normal. A foreign cell has been removed. Now, remember, it's okay for the cytotoxic or killer T cell to have the same mechanism as the natural killer cell because they all originate from lymphocytes. But please keep in mind that your natural killer cell is part of your innate or non-specific defense system, meaning that it will not have a memory of the event or the foreign antigen. Whereas your cytotoxic T cell is part of your adaptive immune system. So it will remember, and it will also have a faster response the next time it becomes exposed to that same antigen. Cells that are part of our innate system, like our natural killer cells, they will always have the same response. Their response will not be quickened or amplified because they don't keep a memory of what's occurring. So on the slide right here, it says cytotoxic T cells and killer T cells will have the same mechanism of killing, but there are some differences, including the fact that natural killer cells will recognize other signs. They will look, for instance, for cells that do not have a class 1 MHC protein complex. They can also look for antibody coating target cells, and they'll look for stress cells that have different surface markers on them. So natural killer cells are able to recognize different categories versus the cytotoxic T cells. The natural killer cells will also have no memory of the event. But then the natural killer cells, because they're part of our non-specific immune system, they also do not have to be primed they don't have to go through the activation step of interacting with the MHC complex and the antigen and the co-stimulatory process. So the natural killer cells will be able to react quickly, but they will not have a memory of the event. Either way, we see that the cooperation or the similarities between the way the natural killer cells and your killer T cells work together is basically an example of what we like to call immune surveillance. Both of these cell types have been known to move around the body looking for markers that they interact with, and they will then swiftly respond to it as well. Now, we've talked about our helper T cells. We've talked about our cytotoxic or killer T cells. Let's also talk about our suppressor cells, T cells, which are also called our regulatory T cells. The regulatory / suppressor T cells are going to be essential because it's going to help us prevent autoimmune diseases from occurring by basically tapering the reaction of the immune system as we start lowering the amount of foreign antigen that it's exposed to. So on your slide I wrote down, it will dampen the immune response by direct contact or by secreting inhibitory cytokines and transforming growth factor beta. So this is all in an effort to prevent an autoimmune disease from occurring. It will suppress self-reactive lymphocytes, and there's doing a lot of research to figure out how they can be used in accepting transplanted tissue by inducing tolerance in our immune cells. Now, the reason your suppressor T cells is so important is because it's able to basically limit the activation of your cytotoxic or killer T cells. Because if they are activated for too long, then what we see happening is that there's an increased chance that they might start attacking self-antigens. If the foreign antigens start to taper down and there's nothing to react with, they might start reacting with self-antigens that look very similar, which of course can cause a host of other diseases and symptoms to appear. Now, I want to go ahead and take a second and show you this figure. This is figure 21.20, and it basically takes most of part 2 and part 3, and it just puts it in an illustrated example. So I think it's an excellent figure to refer back to, and at the end of studying this chapter, if you come back to this figure and you're able to understand the different steps, then you're in a really good spot for the upcoming exam. So let's just quickly work our way through it. Basically, it says that if you have a foreign antigen that is exposed within the body, you can have an adaptive response or an innate response. The innate response is one that's going to be equal for all of us. So it's going to be things like the surface barriers and our internal defenses. So they drew out the complement systems right here. But this is also where you would find your fever, your inflammation, your macrophages, your natural killer cells. Now, a good thing about the innate defense is that it's always ready to go. You always have these defenses ready to be utilized. There's no priming or activation needed. A bad thing or a negative aspect about the innate defense is that it doesn't form a memory. So that means that you'll never have an amplified response. You're always going to have the same steps, the same response level, because it doesn't recognize the foreign antigens that it previously had contact with. On the other hand, you have your adaptive defense system, and that is all going to be about your B cells and your T cells. So on the chart in the orange section right here, it shows you how you utilize your antigen presenting cell, your little dendritic cell, to go ahead and activate your naive CD8 and CD4 cells, and your CD4 cells will go ahead and interact, or I should say will mature and become activated, and eventually will form your helper T cell, your suppressor T cell, as well as your memory T cell, whereas your CD8 naive cell will eventually become your cytotoxic T cell, which can directly kill the foreign antigen, and we're also going to have some memory T cells that are formed. Now back to my helper T cell. I'm going to notice that there are some lines that are drawing it into the blue section of the chart, and that blue section is all about the fact that you also in your adaptive defense system have the production or the activation of your B cells. So I have my antigens interacting with my B cells, and once the B cells are activated, they will go ahead and proliferate, and they'll give rise to my plasma cells, which are my effector cells that are able to secrete antibodies, and also some memory cells. And the reason that the lines are connecting the blue and the peach between the helper T cell and the B cells all has to do with the fact that they can co-stimulate each other, and by co-stimulating, your helper T cell actually allows your B cell to have a more amplified response in its production of its plasma cells. So the helper T cell is the connector of your adaptive system between the T and the B cells, and also partially our innate system. Now, what is the positive and the negative part of our adaptive defense system? The positive part or the benefit of it has to be the fact that it forms a memory. If you have the memory, you can respond quicker and more amplified. So that's fantastic. The negative part of it is that it has to be primed first. So it will take a few days for a new antigen to cause a specific T cell or a specific B cell to become activated. So that, of course, is a negative aspect. So as you can see, the positive in one system is the negative in the other system, and vice versa. Now, one last notation that I want to make about our immune system is the fact that we come across things where there could be some mistakes. And this is then where you can get immunodeficiencies. Immunodeficiencies is basically when you're either born congenital or you acquire a condition that impairs the production of your immune cells. If you come, for instance, across a disease called severe combined immunodeficiency syndrome, this one is a genetic disorder where your body is not able to produce active B cells or T cells. This basically means that you're severely immunocompromised and you have no specific defense signal, which means that you have to almost live in a bubble. In fact, that's where the boy in the bubble came from that we're going to be discussing in our next chapter. It's based on the fact that the SCID, SCID disease causes a defective ADA enzyme, and this allows for metabolites to build up that essentially kill off your T cells. And it can be very fatal if untreated, but unfortunately the most effective method of treatment will be a bone marrow transplant. Why? Well, because remember, all white blood cells are produced by the red bone marrow, and it's from there that they'll then go ahead over to the thymus or stay in the bone marrow for them to become competent and be able to recognize a self-antigen and a foreign antigen. So here on the side, I have a little picture of David Vedder, and if you've ever seen the movie The Boy in the Plastic Bubble, I don't know if that's the actual title of the movie. Maybe you saw the Seinfeld episode where George went to go visit a boy that was living in a bubble. All those storylines are actually based on David Vedder's life. He was born with SCID, so he wasn't able to produce the T cells and the B cells. And basically they had to protect him from himself and his environment because his body only had his innate immune system to protect itself. So he literally lived in a bubble, and all his food, everything that came into contact was completely purified and sanitized before he was able to interact with it. And when he got a little bit older and he wanted to obviously go outside, NASA decided that they were going to build him a special spacesuit that would protect his body from exposure to the outside elements and also allow him to have like purified air so he can go and like see what his front yard and his backyard and all the other stuff look like. So here's a little picture of him outside. Now, when he was I believe 10 or 11, they decided that they were going to go ahead and do a bone marrow transplant on him and see if they would be able to regenerate the T and the B cells utilizing the donor's bone marrow tissue. And it was decided that they were going to use his sister as a donor. His sister was thoroughly tested. They went ahead and they did the surgery, they did the bone marrow transplant, and everything seemed to be going well until a few weeks later. Unfortunately, David got very sick. And he got sick to the point that unfortunately he did lose his life. And when they went to go figure out what exactly went wrong, it turned out that the sister's bone marrow had a small, minute amount of viral particle in it. It was so tiny that it wasn't able to be tested or be picked up by the diagnostic tests that they did, meaning that her body had no problem keeping it at bay and keeping it intact. But David's body, because it absolutely did not have any T or B cells, was not able to fight off that little bit of viral particle, and the viral particle quickly replicated and took over his body. So unfortunately, that then caused his death to occur. David feather is often mentioned when you talk about your immune system because it really does highlight the importance of needing both our innate and our specific systems to work together to give us that overall immune system bubble so that we can live our day-to-day lives as normal as possible. Now, a few more notations. If you come across the term autoimmune disease, that's usually when the immune system makes a mistake and it starts attacking your self-antigen or your own cells. And this can be due to the fact that you can have what they call a cross-reactivity. Cross-reactivity means that you have a foreign antigen that looks very similar to your self-antigen. So your immune system makes a mistake and not only attacks the foreign cells, but it also attacks your own cells. In fact, a lot of researchers believe that most cases of type 1 diabetes, when the pancreas just stops producing insulin, are caused by a cross-reactivity of an autoimmune disease, where there is a certain virus that our immune system is reacting to that looks very similar to our little pancreatic cells that produce insulin for us. You can also have abnormal exposure of a self-antigen. Some of our cells are hidden behind a blood barrier. If something happens to that blood barriers and those cells are able to be exposed to the rest of the body, it can cause the immune system to start attacking those cells, not being able to recognize them because they've never seen them before. Also, mutations such as cancer can cause a change in the structure of your self-antigen, thereby making it no longer recognizable and being treated as a foreign antigen. Autoimmunity is then going to be the production of antibodies that are labeled as autoantibodies, meaning they're attacking your own healthy cells, okay? So this happens in cases like lupus, diabetes type 1, arthritis, as well as multiple sclerosis. So this is when your body is producing autoantibodies, antibodies against your own cells. Now, for many of us, we have probably also been exposed to things like allergies. And if you have allergies, like for instance, if you get hives or even asthma, which is the most common allergy, this is an example of what we call hypersensitivities. And this is seen when the immune system literally overreacts to a small threat and causes massive amount of tissue damage. There are different categories of allergies. It will mostly depend on how long it takes for the reaction to occur and show symptoms in the body, as well as if it involves antibodies or T cells. If it causes the production of antibodies, usually it's more of like a subacute or a medium, a short-term hypersensitivity. If the T cells become involved, then usually it's more of a delayed hypersensitivity. Your hypersensitivities have many differences and there's many different categories. The most common, as I mentioned to you before, is asthma. You also have some intermediates, like for instance, if you have the haptens, that can induce an immune response. So like you start to get allergic to things like perfumes or cosmetics, or detergents. Also, you can have disease models like we were talking about. Those are going to be more of the delayed type. So when you take a look at like arthritis or like lupus. Either way, usually with hypersensitivity, one of the first things that's seen is that your body releases massive amounts of histamine. And as we've talked about before, histamine is going to be a very potent inflammatory inducer. And that, of course, will set off the innate reaction, which will eventually either trigger your B cells or your T cells. I hope you have enjoyed our immune system chapter. Obviously, if you have any questions or concerns, please let me know. This chapter has a lot of different elements, a lot of different names, a lot of different pieces. So take your time, go through it, and keep track of things that you're not really sure about. And feel free to email me at any time, and I'll be more than happy to give you an explanation or some additional examples. All right, everyone, we shall talk soon.